Alright, last but by no means least, quick overview of what we like to call the electron transport chain. Alright, first things first, we are inside the mitochondria. So here's a mitochondria. And we are in the mitochondria you have all these little membranes, and these are called cristae. Alright, when you look at these, there's tons of them. What you are seeing right here is one small section. Let me get a different color. Alright, you're seeing one small section of this. This is all we're showing you right there. Okay? So what the deal is that we're going to see what happens with all those NADHs and FADHs that we made in the Krebs cycle. So I'm going to try and get rid of this, and we'll have more room. Alright, so what the deal is, let me read this toolbar here. You are looking at what are called transports, these things right here. Not important that you necessarily, there's more to it than that, it doesn't really matter. But what we want to do is to say, okay, we are in the matrix, we made NADH, remember? What it does, if you'll remember your cell communication, the surface of this cell on the outside has a part of cell communication. It has a receptor area that allows NADH to fit right there. What happens is that these hydrogen ions, also called protons, are released, and you are left with NAD+. Now that should sound familiar, because that is what entered the reaction not only in glycolysis but the Krebs cycle. What happens is these hydrogen ions move across to this inner membrane space. It's a double layer membrane, just kind of like the cell membrane. Let me remind you too that if you haven't done it yet, that part of this is that mitochondria, they believed, was part of the endosymbiosis. In other words, a cell engulfed another cell. And so that mitochondria are kind of really different. They have their own DNA and everything. Evolutionarily speaking, it's the belief that that's where multicellular organisms came in. And so that they have a double layered membrane, just like your own cell membrane does. Anyway, so we just brought some hydrogen ions in. How did we bring them in? Because electrons were released into this. So you'll notice there's my electrons. They have a negative charge. They pull that positive charge over, and there we are. Alright, the electrons are now going to do a little bit more work. So they're ferried across, and they're carried across by these carrier molecules. Now this one is actually called CoQ. Oops. Alright, and you can buy this one in the grocery store. It's called coenzyme Q. More about that in a minute. So the electrons literally move from here to here. Now, we've already said they have a charge. So they're going to pull more hydrogen ions across. Apparently, I have a hard time making a plus sign. Alright, then another carrier molecule called cytochrome C carries the electrons over to this transport, and more hydrogen ions come across. Also known as protons, better plus signs. Alright, these electrons have done quite a bit of work. Think about that. They've carried across at this point 3 sets of hydrogen ions or protons, and they fall. Now you know that when they fall, that means they're out of energy. They have to be picked up right here, because otherwise nobody else can come and move along. So what happens is that you have oxygen come in here. It picks up the electrons. Now oxygen has this negative charge, and so that oxygen is going to pick up a positively charged hydrogen proton. And what are you seeing? And you should know by now, of course, this is water. So what oxygen is called is, and I'm going to have a hard time writing this one, so we'll try real careful, the final electron acceptor. We'll come back to that in just a second. FAD actually enters, FADH enters in a different place. It comes to this particular transport. It drops off the electrons here, and then hydrogen are pulled across. They jump over here and hydrogen are pulled across. You will notice at this point, let me get a different color. At this point, FADH is only going to get you 2 pairs of the hydrogen ions, whereas NADH got you 3 pairs. That's going to be important here in just a second. Alright, anyway, so back to this for a second. So what we have done is we have put FADH in here, and of course what comes out? FAD plus. Both FAD plus and NAD plus are going to go back to the matrix. Of course they're in the matrix. And they're going to go to the Krebs cycle and they're going to pick up more stuff. That's where that comes from. Over and over and over again. Alright, at this point when you look at this, just remember we dropped off the energy that we put into the bonds of NADH. That energy pulled across these hydrogen ions. We dropped off the energy in the form of electrons that we stored in FADH2, and that energy, or those electrons, pulled across hydrogen ions. And they were able to pull across some more hydrogen ions. At this point, oxygen, which you breathe in, is your final electron acceptor. At this point, while it's getting charged up and stuff, it's called a free radical. You may have heard of these because free radicals can go and pull molecules away from important things that you need and can cause havoc in the body. So you want to try and use your oxygen as efficiently as you can in this step right here to turn it into H2O, because you don't want free radicals running loose. Reason I said remember coenzyme Q, you can buy this in the grocery store. Let's say, for example, that you do not have very much, I'm going to use yellow so it won't be real dark, or no, green I guess. Let's say you don't have a whole lot of that. What are the electrons going to do? Well, they don't jump. So if you take coenzyme Q and you were to literally flood that cell with that, and you've got more of it, see, then you can pass your electrons off, and therefore you can actually get more oxygen used up and you don't have as many free radicals. So coenzyme Q as a supplement actually does help you use your oxygen. Alright, so what did we do all this for? What we did is we want to these particular molecules right here, we want these to to go down this thing right here, which is called, and I'm going to try ATP synthase. It's an enzyme that's going to make, I'm not going to write it all out, synthase. It's going to make ATP. So what happens here is that ADP comes in, and as these hydrogen ions come down this synthase molecule, what's happening is it's going to make ADP pick up a phosphate group, and of course what are we making? ATP. So every 2 pairs of hydrogen ions, protons, makes 1 ATP. Remember that for a little bit later. Alright, one of the other things about this also is how do these things know, let me get a different color here, how do these things know to go down, there we go, this thing? What happens is, if you'll notice, we had some hydrogen ions over here, but we've built up what is called at this point an electrical gradient. Alright, and we'll try this time and pick up yellow. Nope, it's making me do green. There we go. Alright, so like lightning, not great on the yellow, you're getting an electrical charge. Well, your cell doesn't like that. So it has to even it out by allowing those those hydrogen ions to go down this synthase molecule, and that powers, quote unquote, since it's electrons and protons, it's powering this cycle of making ATP. Okay? This process, by the way, besides electron transport chain, it is often referred to in your book as oxidative phosphorylation. In other words, we're going to put a phosphate group on something. That is the whole point of this. And of course, you saw my drawing earlier of the mitochondria that has all of those membranes in it. And this is happening throughout that whole mitochondria, and then you've got tons of mitochondria, and that's how you are going to make your energy. The last thing about this would be, let me go back and let's see if I know how to do this. Edit and clear. And that would not be good because my whole image went away. Alright, I'm going to have to do it this way. All right, what the deal is that let's say you stop breathing, okay? So right here, no oxygen. These electrons get stuck. These get stuck. These get stuck. NADH comes knocking on the door and it can't come in, so it can't drop off any hydrogen ions. And this whole process backs up. It takes quite some time because you usually have a stockpile of ATP. However, eventually you run out of ATP, and that is one of the reasons that you will hear that everybody dies of lack of oxygen from the brain, because the heart muscle, which needs ATP to pump, stops pumping blood. It just takes a while for that to occur. 5 minutes they can bring you back and you might be okay, and then you get into the range of 10 minutes and that's brain death, and then that's not good. And so the electron transport chain is what supplies us with all of our energy. I'm going to stop at this point, but we're going to go back and account for all of your total energy products and how they made ATP. That should help a little bit as far as making you understand why we did all this.